We are just down the road from the Salk Institute in La Jolla, California, at the Estancia La Jolla Spa and Hotel with Terry Sejnowski, who's Francis Crick Professor and head of the Computational Neurobiology Lab at the Salk, head of the Institute for Neural Computation over at UCSD, and a Howard Hughes Medical Institute investigator, and one of only 10 living individuals, I think, who are members of all three national institutes. So it's great to have you here. There is an editorial on the 4th November issue of Science, which you did with Sidney Brenner, and it's called Understanding the Human Brain. And I would like to know, first, give me a quick summary of this. Why now? Why did you feel it was important to do this at this point?
Well, every year the Society for Neuroscience has a major annual meeting. This year it'll be in Washington, D.C., and 35,000 neuroscientists and exhibitors converge and exchange the latest results and advances in neuroscience. And it's hard to even comprehend how much is being done. And every year Science magazine has a special issue timed for that meeting. And this was a guest editorial. Sidney Brenner is a senior fellow in the Crick-Jacobs Center for Theoretical and Computational Biology that I direct, and we have had a dialogue over the last 5 years about many of these issues, and it seemed like a good time for us to sort of write down some of the ideas that we had.
So what are some of the ideas? I mean, the first paragraph is I see some very Sidney phrases in here where he's less than happy about a number of what he would call the omic sciences, connectomics, synaptomics, and all the omic sciences, which seem to be splitting the field into ever smaller pieces. I know that's not the intent, that eventually it'll all come together again, perhaps, but could you speak to that?
Well, it's clear that, and we do say that the omic sciences are absolutely necessary. There's no doubt that having the human genome was a great advance, but one that didn't immediately lead to any immediate insight or curing any diseases, but rather served as the foundation for all the work that's been done since then. It's basically meant that instead of having to go and clone a gene, all you have to do is go to a database and then search for it and then compare it to other genes. And so it's really changing the way people do science. But there's still all the hard work that needs to be done in terms of getting ideas, testing ideas, but now with a much broader database. And I think he makes it an important distinction, and this is something that we had great long discussions about, and the distinction between knowledge and understanding. In other words, having a catalog is knowledge. Being able to say, how every say protein interacts with another protein or binds to DNA is knowledge. However, that doesn't necessarily mean you've understood a signaling pathway, you have understood how a neuron works or encodes information. That requires synthesizing all of that knowledge into some higher form. And in particular case of the research that I do myself, it's putting that into a model system that allows you to be able to understand how the parts interact with each other and what sort of complex behaviors emerge from those interactions.
So you talk here about all the various sciences now which have the prefix neuro, like we have neuroeconomics, neuroethics, neurophilosophy, and so on and so forth. Just speak to the notion of whether that's surprising or not, because a lot of people are worried by it. I think they sense this reductionist notion. I mean, you're Francis Crick Professor, Francis himself in The Astonishing Hypothesis. Do you remember that quote of his?
Which quote was that?
The one about that the astonishing that everything you do, your emotions and so on.
Oh yes, of course. Yeah, but
simply you're nothing but a pack of neurons.
But as you know, he spent the last part of his scientific career studying consciousness, which is on the opposite spectrum of reductionist science, but trying to understand how all of the complex aspects of visual awareness were a consequence of the interactions between all the neurons in the visual system. And one of the things that Francis, I think, put an enormous amount of time and effort into was studying visual neuroanatomy, which has been reinvented in the form of connectomics. That's the new word for understanding the wiring diagram of the brain. And it's clear that knowing that is very important. But again, it may be necessary, but it's not going to be sufficient for understanding the function and understand how visual awareness arises from those networks.
How do you reassure, when you give public talks and so on and so forth, presumably people come up to you and they're interested to know how this kind of thing can possibly, how these kind of sciences can possibly explain individual differences. People as individuals, people with taste and qualia, people want to have free will and so on and so forth. Those are really tricky topics to.
Well, those are all fascinating topics, and they're ones that are at the forefront right now of, I think, debate. For example, neuroethics and how that impacts on our judicial system. There's just a lot of very difficult questions that are going to be.
Yeah, I mean, this is nothing special. This is just happens to be this month's. Some magazines or this. So there's a piece in here about dreaming and another piece about free will. This is Nature for September, the thinking machine, Neuroscience and the Mystery of Free Will. Here, one of the critiques in today's New York Times of the notion that neuroscience can explain everything. There's this whole notion and exchange between Alex Rosenberg and William Egginton, Bodies in Motion, on the blog and on the New York Times. And there's a comment here from one of the people who's basically saying, let me see if I can find it. He's basically saying that you can't possibly reduce things. Considering this, consider this, you're studying a man's brain, you see some nerves light up. What did that man think? You have to ask him in order to find out. In this, the subject identify the object, thus make it inherently subjective approach to learning what certain endings can do. Well, if you pick up The Economist and look in The Economist at this piece here, mind-goggling, it is now possible to scan someone's brain and get a reasonable idea of what is going through his mind.
Well, I think this is the beginning of a new era where, in fact, we will be able to peer into people's minds. I don't think we necessarily will be able to decipher the details of their thoughts, but certainly in broad outline we'll be able to understand whether they're having imagining an image. In fact, this is one of the articles that was quoted there, which was really quite remarkable.
From Current Biology, I think, yeah.
Yeah. So. I want to go back to this fundamental distinction between knowledge and understanding. So neuroscientists have unearthed an enormous amount of information, details about how the brain is organized and how all the little neurons in different parts of the brain. Just in the retina itself, there's 100 different types of neurons, and that's one of the simpler circuits. So we've made a lot of progress because we now have names for all those neurons, and we know pretty much how they're connected. I mean, there's some details we don't know yet, but we're almost there. But that still doesn't equate to a complete understanding of the function of the retina and what all those different neurons are doing. There are 24 Amerkin cells. We have only the faintest idea of maybe how one or two of them are integrated into the circuit. We know. But the point, though, is that there's still a lot of work to do. Mm-hmm. And if you think about it from the perspective of we're really good at taking things apart. So, when you took apart your father's watch, I don't know if you've ever done that, but I remember doing that. It's a lot more difficult to put it back together. And in a sense, having a computer model, which is something that I work on, allows you to do that in the computer. It's a way of synthesizing all of the pieces and trying to integrate them together. And it's what a biochemist would do when they reconstitute the function of a biochemical system to prove that they actually have all the pieces, that they haven't left anything out. They purify each piece, and then they mix them back in the test tube, and they see whether or not the function has been reconstituted. And in a sense, by having a computer model that takes all those pieces and connects them up in the way that we think they're connected in the brain, and by giving them properties that neurons have, and by giving them the right inputs, and seeing whether or not we can recapitulate and reproduce some of the output patterns and the behaviors coming out of those networks, I think that will give us a little bit of confidence that we actually are beginning to understand the circuits.
I think one of the panels, as I recall, that comes up at the Society for Neuroscience meeting is about neuroscience and the law. And again, that's a very tricky area. I mean, how much can you actually import neuroscientific findings into the courtroom? Is this something you've also thought about? Because we've done this in the Beyond Belief meetings as well. This is your Brain on Law, remember? That's
true. It's a hot topic. I think that there's going to be a real revolution because, and by the way, it's not just neuroscience, it's also cognitive science as well, and psychology. These are all different perspectives on the brain, the behavior of the brain, how the brain is, it develops, how does it evolve. Those are all important parts. And just to give you one example, we've already known now for quite a while that eyewitness testimony is very unreliable, and psychologists have figured out a lot of the things that bias witnesses. Just the way evidence is presented can bias a witness. And of course, lawyers have known this all along because that's how they ply their trade. But we're beginning to understand why the brain is fallible to these distortions, and so once we've understood that, we might be able to try to prevent biases from happening, at least the ones that we're aware of.
Yeah, I'm confused. There's this sort of dichotomous position here in the sense that if you take something like education, recent Scientific American piece, How to Build a Better Learner, that's a phrase we've been using at the Temple Dynamics of Learning Center here at UCSD. It's something we've been using, you and I and Paula Talal, with the X Prize Foundation in terms of a neuroeducation X Prize. And the basic thesis is that instead of complaining about bad school teachers or school boards, you say, what can you do to build a better learner? And the answer is to optimize the 3 pounds of wonder tissue you've got up here. Obviously, it's to do with the brain, right? So that's clear, although it's situated in the body as well. So people are very enthusiastic about that, and they clearly sense that neuroscience has something to offer. But then they also balk at the neuroscience intruding too much into their own control of the situation. So this is just a matter of time, you think, of getting used to it? Because if you look at the bookshelves now, there's dozens of books out there in which how brain science can explain fill in the blank. So there's plainly a movement going on, but there's unease at the same time, I think.
Well, there's a lot of excitement, and as we do make progress, there's no doubt that it's going to have implications for the classroom. However, it's not going to be a direct connection. There's much more to classrooms that we've learned from TDLC than just stuffing facts into a brain. There's social interactions that are occurring, social interactions between students, social interactions with the teacher, enormously complicated dynamics. And if we want to really understand the brain, we need to understand it in the context, both of the classroom, of the parents, the family, the home, and those are very, very difficult laboratories. You can't go into a home and instrument it and try to collect all the data that you would really like to have, because that might give you some hint as to how different home lives may influence different brains to learn or not learn. And we're going to have to do that. At some point, we're going to have to bite the bullet. What's really exciting right now is the fact that social media is collecting an enormous amount of data about people's preferences and how they interact with each other. People are carrying around very powerful computers that can monitor the environments that they're in, where they are, how they spend their time. It should be possible in the near future really to actually study education in a scientific way, in a way that hasn't been possible up until recently, because it was very expensive and very difficult.
So I saw a paper very recently. There was one of the authors was Geraint Rees, okay, and it was in the Proceedings of the Royal Society of London. And if I'm remembering correctly, it was basically saying that depending upon your Facebook number, your FBN, the higher the number that you had, the more connections, more friends, that was producing distinct changes in certain brain areas. There was a greater density in certain brain areas. I mean, do you find that understandable? It's not surprising?
We know, and we have known for a long time, that the experience that you have leaves an imprint in the brain, and it in a very direct way, and it can changes synapses. Recently, we discovered that learning can change the volume of white matter, which are the connections between neurons, not the actual synapses that make the connection, but rather the long wires that course through the brain, and 80% of the cortex really is white matter. It's not the thin gray layer that everyone identifies when they see the outside of the brain. And that means that the signals that are going back and forth across those connections are also, in a sense, adapting to the experiences that you've had. So, I wouldn't be surprised if there was strong correlations. Now, understanding the causal connection is something that we don't understand. What was it about that Facebook experience that caused that particular area or that particular, change that was registered? In fact, the more we look at the brain, the more we realize how dynamic it is. Mm-hmm. It's not just a static piece of hardware sitting there with information flowing through and being stored. Rather, it's actually is being constantly reorganized. All of that information is constantly being reorganized. Every time you recall a memory from the past, you've modified it. And reconsolidation, this is a very, very important topic, especially for false memories that have been implanted in people, that things that never happened but make you feel as if they did because you've been told about them enough times so that you really feel that you had that experience. So we really need, this is what we're getting to right now, is to understand if there is so much dynamism, then the question becomes, how is there any stability? How is it that you can have memories that last a lifetime, given the fact that synapses are turning over, there's tremendous amount of the trafficking within neurons in terms of where the resources are going, and that's really exciting because I think we're going to have some surprises.
Well, yeah, but slide off, friend. But let's follow that one because that really I've asked a lot of people on the Science Network as we've gone through Joe Ledoux, Liz Phelps, and so on. And this is a conundrum, as you said. There's this turnover going on. If when you access a memory, you're effectively changing the memory, how can you, at the end of your life, have any reliable sense, any sense that the memories that you have of your childhood are in any way reliable? Why hasn't everything been changed? What mechanism could possibly allow that kind of turnover to be happening, and yet you still have some stability as a coherent individual over time?
Well, the structural memory is probably the longest lasting. For example, your face. As you get older, it changes in a very predictable way. But there are features of your face that you could recognize throughout your entire life. That there's a certain structural organization, and that structure, in the sense of bones and tissue and so forth, that has a memory. But my best example, in terms of an analogy, to try to understand what might be going on in terms of long-term memory, is I'll bet there's a scar on your body that is almost as old as you are. Mm-hmm. Right? And when scars heal, they leave behind some tissue. There you go. They leave behind keratin and collagen, and it just stays. Okay, it's very, very tough, and it's there for your lifetime. So it may well be there is material like that somewhere, perhaps at the synapses. Maybe even what's called the extracellular matrix, which is the glycoproteins that actually bind together neurons at synapses, that those form very, very tight, long-lasting, very, very tough to dissolve scars inside of your brain that could be the source of your long-term memories. Now, that's just a speculation, but that's where I would put my money if I had to bet on the actual structural foundation for where those memories are stored in terms of the actual material. Now, you can. It's quite fascinating to think about this because you can cool someone's brain down, and it happens right when people are in comas. So there's no activity. There's no electrical activity. There's still metabolic activity, but the person comes back and they can recall the past, and maybe they have some retrograde amnesia. But it's amazing that all of that has to be somewhere in the structure of the brain.
So just to summarize at this point, and as you go off to the Society for Neuroscience meeting, what are the things that you're most excited about, or you and Sydney together, in terms of which areas of research are you particularly excited about in terms of the neuroscience?
Well, I am very excited about the potential for studying the human brain. In fact, that's the title of the article. So for most of the history of neuroscience, the only way that you could study the human brain was really following lesions or genetic diseases, disorders, epilepsy, and that's a very crude way of trying to approach the human brain. One of the things that Francis was very concerned about was that most of our anatomy is based not on humans, but on animal models like monkeys, and to a lesser extent chimpanzees, our nearest relatives, because you couldn't do the invasive experiments on humans to find out where the fiber tracts are located. And so the monkey brain, though, is quite a bit smaller and less complex than the human brain. And now, and for the first time, we are beginning to develop techniques that allow us to do non-invasive track tracing using diffusion tensor imaging. And that's getting better and better. This is using brain imaging techniques, magnetic resonance imaging. There's a tremendous explosion occurring right now in areas that had been abandoned in the 50s, EEG and MEG, so electroencephalography and magnetoencephalography. These are external signals that you can measure at the scalp. And now with powerful computer and new algorithms, such as independent component analysis and multi-beam forming, it's now possible to locate the sources of those signals with a very high temporal resolution. The spatial resolution still isn't as good as you'd like to have it, but coupled with functional magnetic resonance imaging, which has the spatial resolution, it should be possible now to begin to understand the traffic that's occurring between different brain areas. And one of the most exciting things I've heard recently in this area was from Tom Liu in our fMRI center here at UCSD, who said that it's been possible to identify 50 independent networks in the brain that are coupled together, different brain areas that are coupled together, that are processing information, that are very dynamically in flux all the time, including the default resting network, which happens when you're not processing information. So this is really an exciting time, because now we're going to be getting a real global picture of all of the flow of information through different brain areas. Up until the advent of brain imaging, we were stuck recording from one neuron at a time, and there's only so much you can learn from one neuron in terms of what its function. You can tell something about what sensory stimulus it prefers, or maybe what motor output it might be contributing to. But in terms of understanding something as complex as perception or consciousness, you need to have a much more global picture of all the interacting neurons.
But what about some of the experiments, and again, there's some recent papers just came out again, although they've been doing some of this for some time now, Christoph Koch, Itzhak Fried, and so on, recording from...
one neuron at a time. Okay, so there's the same fundamental limitation. You have to understand... Okay, here's an analogy, which I heard recently, which I think it's the way to think about it, is that a cortical column, which contains about 100,000 neurons, is like an orchestra. They're all the neurons are firing and playing a symphony together. In other words, they're not just isolated, independent of each other. They're connected with each other, and so they have a temporal sequence, and they are representing very complex brain states. And up until recently, it's like shoving a telephone pole down, and you're going through all this tissue, and you pick out one instrument from 100,000, and you listen to it and said, "What's the tune? What's the tune?" Well, you'll never figure it out. And then, then the great advance occurred when it was possible to put 2 telephone poles down and to record from 2 neurons. But unfortunately, they're in different columns, and they're playing different tunes. So now you're really confused because they may have nothing to do with each other. So until we can record densely from a large population of neighboring neurons that are all in the same symphony orchestra, we won't have any clue as to how they're working together to form populations of neurons and the space-time patterns of spikes that we'll need to understand the code.
Yeah. Do you foresee a time when you'd have a technique like optogenetics that would be applicable to humans, where you can actually be tracing?
Well, you're a little behind, because optogenetics now is being used on humans. In fact, Carl Deiseroth, who's a psychiatrist, actually developed it because he wanted to go in and be able to selectively stimulate different neurons. And one of the most exciting advances in, at least in the clinical side of mental disorders, has been deep brain stimulation, where again you put a big telephone pole down into some brain area and you just stimulate the bejesus out of it, and it helps in ways that we don't understand. It helps Parkinson's disease, helps with depression. It's a great advance. However, it's very crude. What Carl wants to do is to use optogenetics to selectively target a particular subset of cells in a particular brain area, and then using opto means light, you shine blue light, you can either stimulate or you can suppress the activity, and now you can go in and fine-tune the system and perhaps even alleviate some of these disorders with much more precision.
Yeah. I didn't state that clearly because we actually have a conversation with Carl on the Science Network, so I knew about that part of it. And we also had a conversation with Helen Mayberg. So
I should keep up with the Science Network.
With Helen Mayberg, who of course was
the depression part,
who did the deep brain stimulation as well. But what I was trying to say, and I didn't phrase it properly, was whether those kind of techniques, as they become even more sophisticated, what kind of a picture of brain do you think that they will reveal? What kind of a computational device do you think there will be emerging?
It's I would say if I had the answer to that, I would publish it. If I had some good ideas. No, I do think that we have to start thinking in a different way. In other words, up until now, we've thought about how the brain works from the perspective of single neurons. And it's clear that the real mysteries of the brain, which have to do, and that's taken us a long way, by the way. I mean, there's nothing, we have a whole map now of all the different cortical areas that we didn't have before. But now the question is, how do you get a unified percept out of all that? You have millions of neurons that are activated in different parts of the brain. How do you get the sense of an integrated, visual, auditory, somatosensory, world out there that you can understand in some integrated way, and that's a big mystery, and that's where I think understanding something about global communication between different parts of the brain is going to be key to understanding that. So that means being able to record from lots of neurons from lots of different brain areas in real time online, and then be able to use advanced computational techniques and algorithms, machine learning, to try to sort out from that, what are the signals there that are shifting and changing in time, that are computing, representing, deciding, all the things that we see in terms of reflected in behavior. But we're not there yet. But we have techniques, and they're going to be getting more and more powerful. So I'm confident that we'll be able to make progress.
So, a final question. We talked about the orchestra, the symphony. I actually did a program some years ago called The Time of Our Lives, in which I got the Los Angeles Youth Symphony to metaphorically represent this whole enterprise, and Lalo Schifrin conducted them. But the question then was, who's conducting the orchestra? And in the same way, how do you get an orchestra to perform? I know it was a metaphor, but nevertheless it is doing all this synchrony and so on. How does it do it without a conductor?
Well, I think that if you took a symphony orchestra and had them playing together long enough, my guess is the conductor can walk off and the symphony will go on. Maybe not quite as well in terms of the integration, but he obviously gives a timing signal, and you could replace the conductor with a timing signal, and obviously there's some dynamics he's indicating, but it seems to me once you have a very well-oiled machine that really has sort of internalized all of those dynamics and understands how the neighboring players are going to react, my guess is that you could probably get some pretty good music out of it.
Well, I look forward to hearing about the music that comes out of the Society for Neuroscience meeting next week. And
well, that's a good example of a conductorless symphony.
Indeed. And thanks for talking to us.
You're very welcome.
Great.